Certain genetic conditions in beef cattle that cause genetic defects like curly calf in Angus or historically dwarfism in Hereford breeds. And the best way to manage that is really to make sure you don't mate two carriers together. And a very simple solution in order to achieve that is to use crossbreeding, where you're assured that you're not going to bring together two mutant forms of the gene from the mother and the father. All genetic variation is really based on mutations, and so, for example, coat colour is as a result of a genetic difference between different colour coats animals, and that's not necessarily a genetic mutation, it's just a genetic difference. And so when we start to call it a genetic defect or a condition, it depends the situation you're in as to whether or not it's deleterious. Of course, ones that cause embryonic lethality, I think, are deleterious irrespective of where you are, and they're the ones that we're really targeting to try to ensure that we're not mating carriers of conditions that lead to embryonic mortality, because then of course you don't get a calf that shows up as less fertile animals. When we see genetic conditions showing up in cattle breeds, it tends to be as a result of line breeding or inbreeding, where the mutant form of a particular gene is inherited on both sides of the pedigree. So in other words, there's an ancestor back in the history of the calf that's being produced that has given rise to both the maternal and the paternal line, so that when they come together, the form of the gene that's mutant comes from both mother and father, and consequently the calf is affected with whatever condition that particular mutation leads to. And it can be things like curly calf or potentially embryonic lethals, whereby the calf actually dies in utero and you never see a calf born. So genomic information is being used for a number of different traits. So it's being used to produce genomically enhanced or EPDs. So in other words, increasing the accuracy of EPDs in many breed associations. And there we're looking at a number of different traits, basically the traits that the breed associations have records on. But you can also use DNA information for things like identifying carriers of deleterious genetic conditions, or even just genetic conditions that you want to know whether an animal is a carrier of black, for example, or polled or horned, or those types of conditions. So there's a number of different uses that you can use it for, and it depends what your intent is. And also it can be used actually just for assigning parentage as well, and determining in a multi-sire pasture, for example, with several bulls, which calf was derived from which bull. It's another piece of information that improves the genetic EPDs, the accuracy of the genetic merit estimates of animals, but it's becoming increasingly clear we need to keep phenotyping animals as well in order for us to keep retraining the genomic prediction equations, in other words, the genomic information, so that it stays current with the current generation of animals. And so it's absolutely not a replacement. Its real value is providing more accurate genetic merit estimates when animals are at a young age and have very little phenotypes of their own. Genomic selection really just uses DNA testing technology to look at the natural variation that exists in an animal and make more informed selection decisions. It doesn't alter the DNA in any way, and so it's just really what we've been doing with conventional breeding for 50 years, and so there's no concerns regarding the safety of the use of this technology. It's just really more informed animal breeding. So the research project that I spoke about in my talk has to do with fertility and trying to identify animals that might be carrying recessive alleles that result in an embryonic loss. And the important part of that project is that just because an animal is a carrier of an undesirable allele, it shouldn't be just removed automatically from the breeding population. It's important to, if it's got excellent genetic merit, to use it appropriately and try to avoid carrier-carrier matings, where you might actually have an embryonic loss, but still use the genetics for the other desirable traits that animal has. And in order for that type of wise mating decision to be made, you have to have a selection objective that you're working towards, or an index that you're working towards. And I think it's really important that we use the right index, because for too long we've had indexes that maybe are very focused on carcass or very focused on growth, and really you need a balanced index, so you're not doing single trait selection for one particular attribute, but rather you're making selection decisions that are going for an optimum suite of different traits, because no one trait drives profit, and it doesn't make sense to just have single trait selection. And so it's important to be going in an optimized direction towards improved animals, rather than putting too much emphasis on any one trait. And fertility is a major profit driver, especially in that commercial enterprise, and is one that we don't have very good genetic merit accurate estimates on. And so we're hoping that we'll be able to, with this project, get some sort of information that can help with the selection of animals that are less likely to be carrying these deleterious recessive alleles, and if they do have them, to manage them better in mate selection decisions.